the other university in the world. They have developed some database for the gene, and I'm pretty interested in using them in the future. If any more thing, I also knew some information about other city, so I think it will be quite interesting for the future work.
Right. So thank you very much. Anyone else who has followed and has got some comments on it?
Hello, Professor. I'm just missing some part of the conference, but I actually remember about the Dr. Wei who's he was presented about the pangenomic analysis of gastric cancer, and my knowledge is limited, and but I some key point I was remember that the human reference genome is still incomplete, and the human reference genome did not reflect the complete genomic status of diverse population, and there are 1 to 10% of the sequence were misses, and they using the pangenome to discover the gene present and absent of gastric cancer. I just remember that. And
thank you very much. This is nice. Actually, all the PowerPoint slides, they said that they will be available on their web pages. So in case you're interested later on, you could go and retrieve them. We're not going to do it now. The idea is that you are there, you follow, you get a feeling of what's going on nowadays at the bioinformatics field. That was the idea. And I thought that was of interest to you. So basically what we have to do today, first of all, I will start with some theory. So we are absolutely sure now that you are fresh that it will be clear to you. I have some exercises that you will have to carry out, and then we'll discuss them. And then from last time, you had to do some exercises that we'll see if you had some questions or not, and if you managed to do them. Is this okay with you, the schedule for today? So basically, I'm going back to the Word document that I had handed to you last time. I share it as well. And you had done the first exercise, and I'm at the exercise 4 now. This is what we are going to have a look at today. However, before having a look, this is exercise 4. You'll have to do this exercise 4 and hand it to me eventually. I announce it from now. I'll show you how to do this, so don't worry. If you just have a quick look and you say, we have never done IMGT Quest, IMGT Domain Gap Align, and IMGT Collier de Perle, this is absolutely fine because we have not seen them yet. We'll see them together today. So this is exercise 4 up until the end, 5, 6 as well. That will be your homework. That was the first announcement. And then I stop the sharing and I'll go to the theory that we'll cover today. Give me a second. I open the PowerPoint slide. This is PowerPoint slide 2, but I'm not sure. I have not shared it with you yet, but it doesn't really matter. I will. Right. Do you see my screen? Guys?
Not yet.
no, not yet.
You don't see my screen. All right. Share screen. Now you should be able to see it.
Yes.
Right. So now this, you remember it. We had seen this PowerPoint slide beforehand. It's a bit a reminder for you. We have explored a lot on the genomic level. You have seen at the chromosome level where the V, the D, the J, and the constant genes for the heavy chain. And we had seen for the variable, the joining, and the constant genes for the light chain being either kappa or lambda. And we had said that this huge number of antibodies at any given moment in our body, 2 times 10 to the power of 12, there are 3 different parameters, the potential recombinations, we have covered this part, and there is the mechanism of N diversity and somatic mutations only for the immunoglobulins, not for the T cell receptors. And we have not seen these 2 mechanisms. So today, we are going to focus on these 2 mechanisms. Let me see how can I go. IMGT Quest is a tool, software tool, that we are going to discover and explore today. Imagine that we have a blood sample from either a healthy or a patient. The idea is there is some sequencing being carried out, either DNA or RNA sequencing, and then we want to compare the reads, we call them reads, the sequences, against the genome, against the reference set, that we call it. And here you get an idea of what the IMGT Quest tool does. The sequences that you have sequenced from the sample, the blood sample, they are compared against the reference data set. I repeat, and here you see the different accession numbers and the names. Now I suppose you recognize perfectly well these names. Immunoglobulin. I'm not sure if you see my cursor. Now you might see it. Immunoglobulin, heavy, variable, subgroup 1-3, the first allele, 0, asterisk, 0, 1. You see the sequence that I submitted. This accession number is my own sequence. And then below, you see the sequences that have been retrieved from the database, the IMGT database. And if you pay close attention to it, every time there is a letter, there is a difference between the reads, the sequences from the blood sample against the sequence in the database. So basically here you see that there is a G, whereas in my case there is a C. Whereas when there is a dot, it means that there is identical nucleotide. Is it clear how we read this? This is for the V gene. Equivalently, that is for the J gene. And here at the bottom, maybe this is easier, we have the V gene, we have the J gene. Here there is so-called junction. And I'll describe to you what is the junction with this slide. This is a really important slide. So basically, I'll go through it step by step, so it's clear to everybody. We are at the chromosomal level. We have the full length of the D region, of the D gene. D genes, they're usually short. We have the left part, the 3' of the V region. V genes, they're a bit longer, so there should have been a bit more just for space constraints. I just gave you the 3 part here. Equivalently, I give you the 5' of the J gene, just because once more, for space constraints, it is a bit longer. So I'm not saying anything new here, The only difference is that this time I give you the nucleotide sequences of these genes instead of giving kind of a graphical representation with bars or rectangulars. Is this clear up until now, or do you have any questions?
Clear for me.
Perfect. Any questions? This is quite important, so you understand the two mechanisms. So if, please don't let me go up until then and then tell me I did not understand anything. I go slowly, and I'm happy to repeat as many times as you need up until it's clear to everybody. So I suppose it is clear to everybody that here we are at the genomic, the germline level, and we have the three genes. Look at what happens. It's like Pac-Man. There are enzymes. You're biologists. We are biologists. We understand. There are some enzymes, endonucleases, that they start chopping, matching away some nucleotides. The number of the nucleotides that they're being cut away and their nature, being it purines or pyrimidines, it is not known within literature. And look at what will happen again. There are some nucleotides that they're falling from the sky. Once more, it is random, this process. That is, neither the number nor the nature of these nucleotides is known. And then you might ask me, so this is a waste of energy. From one side, we're chopping nucleotides randomly. From the other side, we are adding nucleotides randomly. Why is this the case? Why would be the case? Why do you think this happens this way? What's the advantage of this procedure? Guys, this is a question. What do you think the enzymes, these endonucleases and the TDT, they chop away nucleotides and then there are nucleotides being added? Why do you think this is the case? Are you with me?
Yes, I'm still figuring.
So I stopped the sharing so I can see you. Did you understand my question, please?
Is that the DNA repair mechanism?
Why would we have this repair mechanism? What do we need this for? Actually, they're not really the repair mechanisms there, but you are asking a good question. What would we need to repair something? Do we need to repair something? And if we need to repair, what would this be? Any ideas? This is important to understand. This is why I'm trying to make you think a bit, so you give me some answer to this before I move on. I'm sure when I give you the answer, you'll say that that was obvious. I'll give you a hint. I'll share again this slide. What did we say? We said that we have a huge number of antibodies at any given moment in our body. Why is this the case? We have a huge number of different antibodies. Why is this the case? Why should we not have one single type of antibodies?
Because each antibody will attach to a certain antigen, and that antibody will not attach to another antigen. So we need the huge number of antibodies.
Perfect. That's the correct answer. In order for our body to be able to fight any different antigen attacking us, we have to have a panoply of different antibodies. And this is a huge number. And one of the questions that we asked ourselves is, how do we come up with this big number? And in order to come up with this big number, we said we have potential recombinations. Remember, we do not have a single variable gene. We have many variable genes, talking about the human genome, but the same holds for the other jawed vertebrates genomes. We have several variable genomes, variable genes, and there will be one selected for the transcript. Every time, there will be a different one, most probably selected. And this is one of the reasons that accounts for this variability. And then I went further down. I go to this slide, if I manage. I would like to make it full screen so I see it again. Stop sharing. Do you see my slide?
Yes.
Yes.
However, you see the final version of it, where I don't know, sometimes it accepts me to put to put it on the full screen and I show you the animation. Sometimes it doesn't. And I would have liked to show you the animation as I was showing to you beforehand. So you see what is happening. So verbally, I'll describe it again. I told you that there are some enzymes called endonucleases that they will chop off some nucleotides. And then I told you there are some enzymes called, there is an enzyme called TDT that they will add randomly nucleotides. And my question to you was, why do you think this is the case? Why do you think there is this energy waste? And I have already given you the answer. Now you see my screen on the full screen, and I hope I can do the animation. I repeat, we have, before the recombination, we have these enzymes that they delete nucleotides, and we have nucleotides that they are being added randomly. And I repeat, when I say randomly, we do not know either the nature of them, nor or the number of them. And my question to you was the following. Why do you think there are these mechanisms in place? What's the advantage of having these mechanisms?
In order to generate a very high diversity of repertoire, and
Perfect.
that
And that, go ahead.
and that will lead to that we will be able to generate in our body a very high number of diversity antibody in our cells to to deal with many different kind of antigen come from a pathogen.
Perfect. Exactly what you said. And then I have a second question. If we delete and add nucleotides randomly, do you think that the open reading frame will always be correct? You know what the open reading frame is, right? For instance, I don't know the genetic code by heart, but you know that 3 nucleotides, this is a codon, and this will code for a specific amino acid, correct? This is prerequisites. We don't cover this in this class, but I think you know about this, correct? Guys?
Yes.
Yes.
You know this, correct? You know that TGT, for instance, codes for a specific amino acids, GCG and other amino acids, and so on and so forth. Now, if we delete nucleotides and add nucleotides randomly, we might have a frame shift. We might have a change in the open reading frame. Is this clear to you, this?
Yes.
Correct. So there will be several of these sequences that they will be aborted. They're not going to give rise to a protein because here there could be a stop codon. There could be 3 nucleotides added that they code for a stop codon. If there is a stop codon, of course, you realize that my peptide will stop here. It will not go any further. If this is the case, this sequence will be aborted. It won't be a proper antibody sequence. And although there is so much waste of energy, in quotations, the waste of energy, this is a really efficient system to generate variability. Look at something more. The story is not over yet. This is what we call the junction. So basically the junction consists, if someone asks you the question, give me the definition of the junction consists of the 3' region of the variable gene, the whole sequence. Sorry, you have a question?
No, sorry.
The junction consists of the 3' region of the variable gene, the whole sequence of the D gene, the 5' region of the J gene, as well as the N region, the nucleotide regions. And what are the N regions? I repeat, they are the regions where we have where the nucleotides have been added randomly. As I said, it's not over yet. Look at what will happen. You see, for instance, look closely at the 3 last adenines here of the V region. What happens? This is the so-called somatic mutations. There are some fine-tuning. There are these antigen-presenting cells, dendritic cells, that they will have a piece of the antigen that they will be presenting it, and then there will be a fine-tuning of the sequence to increase efficiency of the antibody. And these, for instance, these adenines, they are being turned into cystains. And this is the so-called somatic mutations. Sometimes you'll see them somatic hypermutations. This is the junction. These are, so we described at the molecular level 2 additional mechanisms, then diversity, and the somatic mutations that they are found for the moment within literature, it's stated that they are only found within immunoglobulins and not T-cell receptors, I repeat. And this gives the three mechanisms that we talked about that account for the great variability of the antibodies in our body. And what is this? This is a graphical representation of the V region. This is the so-called collier de perle. The French term is used in the literature. And then you see, for instance, remember what are the CDRs? Complementarity determining region 1, complementarity determining region 2, complementarity determining region 3. So we said that they are the three loops that they bind to the antigen. And we said more precisely, the third loop, this one here, is the one which is more variable among the three, and this is the one that mostly gets in contact with the antigen. This one is framework 1, this is framework 2, framework 3, and framework 4. You see some square. What are these squares? These are the anchors. You know what an anchor is? A boat, a big ship has got an anchor to stop it. So basically, these are the anchors of the CDRs. How many anchors do we have? I let you count the anchors. I just go 1 second.
There are three.
How many anchors? Yeah,
There are three anchors.
Where do you see them?
The thought chain.
There is one anchor here.
Yeah.
An anchor here. How many are they?
So there are 6 in total.
6 in total. Well done.
Yeah.
Give me 1 second. I close the window which is next to me before I freeze, and I'll be right back. 1 second. Right. We have 6 anchors. What are these? What sort of amino acids is this P that is in yellow background? Look it up if you don't know. So it stays with you. What is this P as an amino acid? What amino acid is this P? Guys, the question is, what is this P? What amino acid is this P?
The proline.
Correct. And what sort of amino acid is this? Is this a big amino acid? Is this a small amino acid? Which are its physicochemical properties?
They are non-polar, aliphatic.
It's a small non-polar amino acid which allows flexibility. So this is why they are given, and you see several times there are these kind of turns at positions where some flexibility is needed. What is important is to have a look at this 1, 2, 3, 4, 5 amino acids that they are given in red. There is, we talked about the IMGT ontology briefly, and there is this nomenclature that we talked about. There is numerotation, that's called, and the very same term is used in literature. So basically that you see that all the amino acids are numbered. There is always, irrespective of the antibody, at position 23, a cysteine. a position 41, a tryptophan, W. At position 89, there is an hydrophilic amino acid. At position 104, there is a cysteine. And at position 118, there is a phenylalanine. If we are talking about a light chain, and a tryptophan, if we are talking about a heavy chain. You might be asking me, so do we always have these 5 amino acids? Yes, these 5 amino acids, they have been found to be extremely conserved among all amino acids. Therefore, their positions, they have been standardized. However, have a look. We have some amino acids that get out of the way, or we have some other amino acids that they are given here with hashed lines. And you might say, what is this? In order to keep the same numbering for all the antibodies, we have to have some hashed circles. That is, there could be a different antibody that we are looking at, and there would have been an additional amino acid here. Is this clear? Why we have the hashed lines on the amino acids?
Okay.
Okay?
Yes. Okay.
So basically, this is just a graphical representation. And we'll get to see it in real as well. Here, there is a table classifying all the different amino acids. For instance, this proline that we talked about, that was given in yellow in our graphical representation, you see, it is based on the volume of these amino acids. It falls in the small category. Here is the different hydropathy classes. It is a small, neutral, non-polar amino acid. Is this okay?
Yes.
Yes.
Of course, you find several of these tables in any textbook, biochemistry textbook. You can have several of these tables. This is the IMGT established table that reflects more precisely the amino acids and their properties within the immunoglobulins and the T cell receptors. So basically, that was more or less the theory that I wanted to discuss with you. And now I will go online. We'll do it together. I'll go to let me share again my screen. I'll go to the IMGT webpage that you know by heart now, imgt.org. And then I'll go further down where are the different tools. And I'll choose the first one in line, sequence alignment software for immunoglobulin and T cell receptors, IMGT VQuest. I'll click on this one. First of all, for the ones that they prefer reading rather than listening, there are publications here that if you're a researcher and you use this software, you cite them. Or if you're a student and you want to know a bit more, you can go through them. For instance, I don't know, do you see it or not? I open it in a new. Do you see my publication or not?
Yeah, I see.
Right. You can go and read this publication, which was one of the first ones, I think, 2008. Well, maybe not. Yes. It was even older. I think it's one among the first one. So this is the first thing. Then we'll do an example. You see here, human sequences sets to test IMGTV request are available here. So basically, I'll take a set myself. I click here. I take the first sequence. It's a test. So I'll take this one so I can demonstrate to you how to actually, hold on a moment. Before I go back, do you see my page?
Yes.
Set for first time user from human immunoglobulin. All right. And I took the first sequence. Here is in the FASTA format. Do you know what the FASTA format is? There is always the greater than symbol, a description. It is a really good idea to have the accession number. And then there is the sequence given either in nucleotides or in amino acids. In our case, it is in nucleotides, and it has to be in nucleotides because IMGTV Quest is a software tool that works with nucleotides. So I go back here. I choose the species. The sequence I chose was from Homo sapiens. Receptor type or locus, I put the IG. Several times the students, they forget to fill in this information. Doesn't matter. You'll have an error given back from the software tool. Here it's a sequence submission box. Either you type, I'm not going to type all the nucleotides, I'll copy and paste in FASTA or in FASTQ format. In case you do not know or you do not remember what I just said for the FASTA format sequence, you go here and you read it. I paste my sequence, let me see it. This is my sequence, correct. And then I don't display results. I leave what there is by default. We'll have a look at it later on. There is different things that they can be added at the output file. There is the synthesis view, there is an Excel file, there are advanced parameter, advanced functionalities. For the moment, I leave everything, the default values, and I'll start my analysis. And this, you can think of it as a sequence that I sequenced in my lab after a blood sample. There was a patient, I took a blood sample and I sequenced in the lab, and now we want to see at what it corresponds. I submitted my sequence. If you see here at the top of my window, it's running, it's moving here, and I have my results. Thank you for using IMGTV Quest. Once more, the publications, species, it repeats the parameters I gave, Homo sapiens, receptor type IG, IMGT directory reference set. What is this? This is what my sequence is being compared against. It's being compared against the functional, the open-reading frame genes, as well as in frame pseudogenes. And for the advanced parameters, I did not search for insertions and deletions. So there is one sequence. I only gave one sequence, and this is the accession number of the sequence I gave. It gives me some information about the release of IMGTV Quest of this tool. I'll go further down. Hyphens show nucleotide identity, dots represent gaps. We'll get to see it, we'll understand it much more when we see it visually. So once more, it tells me that the sequence I provided has got a length of 678 nucleotides. And this is my sequence. And it has put in green the region that was identified. So this table here, this is the most important table. This, I'll make it a bit bigger. This is a synthesis table. How do we read it? A result summary. This is my accession number. And then I go to the second line. Before, I'll get back to this one, but I start with this one. V gene and allele. So this sequence, I repeat, was compared against all the sequences in the database, and the V gene, the variant gene and allele that was found most closely related to my sequence was immunoglobulin heavy variable 3-30, the fourth allele, and this is a functional sequence. Or, it gives me an OR, the 3-30-3, the third allele, and this is a functional as well. My question is the following to you. How come my sequence has got two variable genes? Which one is the correct? Why does the software provide me two different variable genes? What do you think? Do you understand my question? I'm trying to have some interaction in order to help you as much as I can. And if I don't hear anything, neither yes nor no, I'm not sure if it's a monologue and I talk to myself, or if you are with me and you follow.
Can you repeat the question?
Thank you. Great. Even this is absolutely fine. So basically, we started off with a sequence. This is the sequence. You see my cursor. That was sequenced from a blood test. And this is an antibody sequence. And I would like to know which are the genes that they are behind this sequence. So what do? I take it and I use the IMGT VQuest tool in order to analyze it. And when I analyze it, I said, I compare it with the total number of variable, diversity, joining, and constant genes because it's a human sequence, against the human dataset that they are available within IMGT. And that's what I did at the interface that I showed you beforehand. And now I'm asking you, I see here at the summary table that the V gene, I have two possibilities for variable genes, either this one or this one. And my question is the following: how come that I have two possibilities? How could it be possible that I have two possibilities for the variable gene and not only one? Is this clearer, my question?
Yes.
Perfect. So what could be your answer?
I think when the tool compare our sample to the database, they give like alignment or something like that, so they can calculate the identify between our gene with the others in the database. And I think we have two results here because two variable gene here give the same score identify. But
Exactly. This is exactly what you're saying.
Yes.
There is a comparison And these two alleles, they have exactly the same score. If you see here, there is this the very same score, and the identity percentage is identical between these two alleles. So the tool cannot precisely say which one of the two is the correct one, because there is the very same percentage of identity. Whereas in case of the J of the joining gene, for instance, you see there is only one result. It was clearly identified that the Ig heavy joining for the second allele, which is a functional, was the one within my sequence with identity percentage 91,65. The D gene, the diversity gene, was identified precisely as well. Is this a heavy chain or a light chain that we are looking at? And why?
The heavy chain.
Well done. It's a heavy chain because there is a diversity gene. And the D region is in reading frame 1. Here, this line gives us the framework IMGT lengths, that is framework 1, framework 2, 3, and 4. The CDR IMGT lengths, that is 8, 8, 11 for the first, the second, and third. And then it gives us the junction that we just had a look at the theory of what a junction is. And in case you don't remember, I'll send you the slides and you'll go and revise it. And this is the amino acid sequence of the junction. I'll go back to the very first line and I'll leave the last line and I'll get back to it. There is a productive immunoglobulin heavy rearranged sequence. The sequence that I have provided to be analyzed, there is a term, it could be either productive or unproductive. Productive, it means exactly what there is in this parenthesis. There are no stop codons, and the junction, there is an in-frame junction. If it was an unproductive rearranged sequence, there should have been either a stop codon or the junction would not have been in frame. Okay? I'll go further down to the file. This is your first question that I would like you to understand, and I'll show you all the sources that you can use to understand, because if I describe myself, you'll get lost. If you go and try to understand it yourself, I'm sure that the majority of you will understand, and someone of you will present it to us. I would like to understand, well, junction length in nucleotides, there is nothing to understand here, the 39 nucleotides, and decryption. I would have liked, or I would like you to understand what this means. There is parentheses, there are these curly brackets, and they mean something. And this is the explanation here. It's a bit complicated at first sight, but if you look into it, it is not at all complicated. You need to bring back to mind the little Pac-Man slide that we were having a look at. I'll get to it a bit later on. So basically, I'll go through the output file. This is the alignment for the V gene and allele identification. Here, it shows us the 5 first results. Of course, you could either reduce or increment the number of results you have got. You see, have a look at the identity score of the 2 first results. you see that it's precise the same percentage of identity between the two. And if you remember, at the summary table, we had the first two results shown here. And what your classmate told to us, it was the correct answer. This is our sequence, and this is the alignment that is provided below. Have a look at the first two sequences. They have a cysteine, there is a guanine. So basically, and then you read it the way you would read a book, like from left to right, and then it continues here. And then you see that they are the same differences, the same substitution or whatever you want to call them, between the two first sequences, which gives the same percentage of identity. Whereas, for instance, look at this last one, there is an additional T as a difference. Therefore, the percentage of identity would be lower. I suppose this is clear. Our sequence is a bit longer. It goes, it is longer. This is, I got the very top. This is why this part is given in green and the rest is not colored. This means, from the alignment, you understand it much better. This means that the CDR3 stops here, and the rest of the sequence is something that has not been annotated within IMGT. And this is the alignment of the variable gene. Equivalently, there is the alignment of the J gene, the joining gene, and the allele identification. In this case, although there are 5 results retrieved from the database, there is clearly one among them which has got the best identity score, 91.67%, and this is the one that was given at the synthesis table. You see the alignment here with all the different alleles. I go to number 4. If you have any questions, you stop me. I suppose this should be clear, straightforward. Actually, there is nothing difficult here. I go to number 4, results of IMGT junction analysis. And look at here. How do we, here is, look at this. I'll come back. Here, this is the accession number. Memorize it more or less, AB012909. And if you go to number 4 again, this is the same input. This is our own sequence, the one that we provide. And this is one of the sequences retrieved from the database. This is the 3' of the V region. This is the N1. There is D region. This is the D gene. The N2. This is the 5' of the J region. And this is the J gene that is being used. And this is the D gene that is being used. Now, if we click on this one, the underlined C, it is given the G. And what does it tell us? Click on mutated underlined nucleotide to see the original one. So basically, it shows us what was the original, what was at the chromosomal level, the nucleotide, and how it changed after the rearrangement. This is what we showed graphically at the Pacman and the somatic mutation changes. And this is what exactly we discussed about the N region. And here is the junction decryption. I would like you to go and understand this. I'll get back to you how you can do it. I'll give you a little hint. How many nucleotides do you see here? For instance, count them.
We have 8.
Right. Look at this 8. So basically, this junction decryption, it represents a precise algorithm and its formulation with numbers. It represents how we describe this junction. I would let you understand how this works. There is documentation that tells you exactly how this works, and you can go and understand it. And I move on to the junction, translation of the junction in amino acids. So if you remember, we said that positions 104 and 118, there are 2 positions among the 5 that they are extremely well conserved. At position 104, there is always a cysteine found. And at position 118, there is either a tryptophan or a phenylalanine. In that case, because we are dealing with a heavy chain, we have a tryptophan. If we were dealing with a light chain, we would be having a phenylalanine. These conserved positions are given in red. Now, once more, it tells you click on mutated amino acid to see the original one. Excuse me. I hope I'm not catching any cold. So basically, I can click on the different amino acids and see if it gives me any difference or not. There are some so-called silent mutations. What is a silent mutation? I'll let you think about what it is or look it up on the internet. You give me 1 second, I'll blow my nose, I'll cut so you don't listen and I don't disturb you, and I'll get right back. All right. A silent mutation? I'm sure you all know what a silent mutation is, please.
It is a mutation in the DNA and not effect on the phenotype of the organism.
Perfect. Exactly. And this is of interest because we could see whether a mutation of the nucleotide level of this junction has got an effect or not in the antibody. This is the junction. You see it here, CARTA, CARTA, and then I'm not sure if it works, but I'll try my luck. Here is a physicochemical descriptor. This, I don't know, I don't see anything. Sometimes it does not really work that well. Well, doesn't matter. We'll leave this. It gives all the physicochemical properties of the amino acids. Doesn't matter. And then from 5 onwards, we have the sequence of the junction in nucleotide and amino acid. We have already seen it. This, you might say, why do we repeat them? We repeat them because several users, they would like to have access to the data, and it's an easier way to retrieve them. For instance, this graphical representation, if someone wants to get this junction, it's not always as simple as that. So basically here, it's in the so-called, the nucleotide, it's at the FASTA format, which is the most frequently used format. Here, there is I go to number 6 of my output analysis. It gives me the V region alignment according to the IMGT unique numbering. What is important and interesting to keep in mind here is this one, the framework 1, the CDR1, the framework 2, CDR2, the delimitations, where it starts and where it finishes its region. The framework 3, according to IMGT, and the CDR3. And then I'll give you some theory background now. We'll discuss, I'll stop my sharing, so there will be a mental break as well for you. Have you heard about, we have human antibodies, of course, our own antibodies. And we have, for instance, mouse antibodies, right? Mouse, most of the box. What is most of the box? Mouse antibodies, mus musculus antibodies. Different species, or Ratus norvegicus, rat antibodies. This written it is quite misleading. If you read what I say instead of listening to it, it is not always transcribed properly. And I suppose, especially the scientific terms, I see that they are not given properly. So when I talk about mouse, I'm sure that you all understand what a mouse is, or mus musculus, and then we are not Christmas scholars. And then we are not talking, I'm not looking at it anymore. So basically, we have human antibodies and we have a different species, mouse, rat antibodies. What is the definition of a humanized, and what is the definition of a chimeric antibody? Guys, are you with me?
Yes.
Mm-hmm. Do you know what is a chimeric antibody and what is a humanized antibody? Chimeric antibody is an antibody that has got Fab, the antigen binding region, coming from mouse, and the rest is human. Whereas a humanized antibody, it's an antibody where only the CDR, the 3 CDRs, come from mouse or any other species, and the remaining is a human antibody. What do you think is preferable, a humanized or a chimeric antibody? I'm waiting for your answer.
In terms of antigen binding, I think the chimeric antibody is better. But if in terms of a antibody that's used for a human therapy, I think the humanized antibody is better.
Right. Maybe I'll get back to you with a schematic representation so it's much clearer to you, because the answer, thank you for attempting to answer, but it's not quite the right answer. I'll give you a bit more hints. The mouse has been attacked by an antigen and it has developed a really good antibody. The human is being attacked by the same antigen. Can we take the mouse antibody and throw it into the human body and the human will be protected by this antigen? Yes or no, and why? Yes? Guys, are you with me?
Yes.
Okay. So I'm asking you a question. I repeat the question. There is a mouse antibody which is really efficient against an antigen. The humans have been affected by the same antigen. Can I take the mouse antibody and throw it into the humans? Will it work?
It's not.
I think it's not.
Why?
Because when the mouse antibody goes to the human body, so the human immune system can destroy mouse antibody.
Exactly. It will be aborted. It's the same with the organ transplantation. You gave the right answer. So basically, I give you again the definition of a chimeric antibody. We said that an antibody has got this shape, this Y-shaped, where we have the constant area and we have the antigen binding area. A chimeric antibody is an antibody that has got the constant area human and the chimeric, this antigen binding area, it's from mouse. Whereas a humanized one is an antibody that has got the constant area from human. A lot of this area is human, it's only the 3 CDRs, the 3 little bubbles, the 3 little loops that they are from the mouse sequence. And my previous question was, what do you think is preferable, to have a chimeric or a humanized antibody? And why? Yes? Do you need to think a bit more, or?
And can you paraphrase the question so I more to clear the question so I read the answer?
Yes, I can repeat the question. The question is, do you think that a chimeric or a humanized antibody is preferable to tackle an antigen in the human body? Do you understand what is, in order to answer this question, do you understand what is a chimeric and what is a humanized antibody?
Yes, we know about the structure of the chimeric and the humanized antibody.
So the question is it preferable to have a chimeric or a humanized antibody? And why?
I think humanized antibody is better because we just need CDR to catch the antigen. So we don't need the whole variable from now to attack the antigen.
Yes, and why is it better? Guys, this is correct what you're saying. It's better to have human sequence than to have mouse sequence in us. Otherwise, our immune system might recognize it as something foreign and might attack it. So it might abort it once more. And we don't want this to happen. We just want to take advantage of the efficiency of the mouse antibody. And this efficiency is found within the CDRs, because these are the loops that they bind to the antigen. The remaining of the region, which is in mouse sequence, it is not of interest to us at this stage. Is this clear?
Yes, it's clear.
Okay, thank you. So I go back to what I was showing you, the results. I'll continue sharing my screen. We were here at number 6, and I was talking to you about the unique numbering, and I was telling you that it's of interest to notice the framework and the CDRs, because we might do an exercise towards the end of the week, we'll see, that will have to do with the humanization of the antibodies. And this will need that we have the precise regions of the CDRs and the frameworks. And then at that time, I bet you'll ask me, where do I find them? And this is the answer. Here are the framework, 3, for instance, and here is the CDR3, for instance. Equivalently, at number 7, you have what you had at number 6. However, in that case, you have it in amino acids. This would be of interest to us as well when we explore humanization of an antibody. I'm not going any more into details here. I think it's straightforward. You have the limitations, the start and the end of the frameworks and the CDRs. Here you have at number 8, you have the V-region protein display, framework CDR, framework 2, CDR2, framework 3. Here is a table, number 9, with the different mutations and amino acid changes. How do we read this table? Look, for instance, at, say, which one do you want to have a look at? I take a look at this one. Framework 2, at position 118, there is a cysteine that has been replaced and has been given rise to an adenine. This had an amino acid change from an H, histidine, at position 40, to a T. What's a T as amino acid? Guys? What is the single code of a T as an amino acid?
Threonine.
Well done, threonine. And in this parenthesis, there are 3 symbols. These 3 symbols, they could be either a plus or a minus, and they represent the change of the histidine to a threonine at the 3 different levels, that there is the hydropathy, the volume, and the charge, if there has been a big difference or not. So basically, when we have 3 minus, it means that there has been a big difference between the 2 amino acids. When we have, for instance, 2 plus and 1 minus, it means that they are quite similar. They are in the same, they are cousins, they are in the same family. They share 2 physicochemical properties that they are the same between the 2 amino acids. Number 10, this is, it gives us the V region mutation and the amino acid change statistics. I'm not going to go much into detail into this one. If you want to explore it by yourselves, I think it's straightforward. Same here. It gives us number 11. It gives us the mutation hotspots where we have even more mutations. I'm not going to get into this one. At the end, 12 and onwards, it gives us, for easiness, to facilitate the life of the user, it gives us the V-region nucleotide sequence in FASTA format, the V-region amino acid sequence in FASTA format. In one line, here you get the V-region amino acid sequence on one line. You get the VDJ region nucleotide sequence in FASTA format, and so on. Basically, it gives us all the sequences that we would possibly be interested in FASTA format and according to the numbering, IMGT numbering. Here, it provides us the annotation by an internal software tool named IMGT Automat. We had a look at it at the previous class within LeakDB, where we had seen, for instance, the V region, and we had seen all the information provided here. That was the exercise that you did more or less for the constant region, the one that you had to do it for today, where it tells you, for instance, framework one is from 1 to 75. The first cysteine is at this precise position, and so on. And at the end of this detailed annotation, you had the sequence, and you had to go and find it. This is the work of the IMGT biocurators. Number 14, it's the IMGT Collier de Perle. I'll click on it so we have a look. We have a look at it. Once more, IMGT Collier de Perle is a software tool that gives us the possibility to visualize the graphical representation of this region, this variable region, for instance. And this is what we are looking at now. Maybe I make it a bit shorter and I get this out of the way so we can look at it more clearly. Here are CDR1, CDR2, CDR3, as we said, the 5 conserved amino acids, cysteine, tryptophan, hydrophobic, cysteine, tryptophan because we are at the heavy chain. We have the different prolines. What is this, the blue color? What is the blue background? What do you think this is? I'll give it as an open question and I'll get back to you and ask you what you have to figure out. This is the anchors, the squares, the 6 squares, the arrows that we see, because they're beta sheets. You know, in the structural biology, there are alpha helices, beta sheets, and loops and terms. These are beta sheets. It gives either it comes towards us or it goes away from us. It gives how they are arranged in the space for the ones who can visualize this in the space. And I go back. And that was kind of a detailed description of an IMGT request analysis. Is this clear? I suppose
yes. Okay.
it is one thing that I showed you all of this, I went through and this and that. It's a different thing that you do the exercise yourself. So basically, I would like what I told you at the very, very beginning. I would like that, where is this? There you are. I'll go through and share it. You have the Word file. I'll share it again. This is the exercises that I would like you to carry out and hand back to me. So I'll go through so you are sure that you understand what to do. You have 3, they are being given 3 rearranged sequences to you. You have the accession numbers. You should be able to retrieve the accession numbers, either from LickDB. It takes a bit of time, LickDB, to retrieve the sequence, or you can retrieve it from publicly available databases. The accession number, I repeat, is a unique identifier. It's like your passport number or your ID number. You'll use IMGT request, and then I would like you to answer these questions. What is the name of the V, the T, and the J gene, which is the most closely related to your sequence? I have given you even hints. Okay, it's in French, but it doesn't really mind. You use Google Translate and you find it in English if you don't have any French knowledge, but I think you might have some French knowledge as well. Which is the functionality of your rearranged sequence? We said it could be either productive or unproductive. It tells you to explain. You'll have a quick look at it and you'll realize if they're stop codons or if they're in frame or out of frame junctions. You give the length of the CDR, which will you find in the result summary. For specifically this sequence, it asks you, which is the mutation rate? If the identity rate is 90%, for instance, the mutation rate is 10%. And then there is a question, which is the prognostic of the patients that have got this CLL, chronic lymphomatic leukemia, if they have this percentage rate? In order to answer this question, you have to search a bit into the different publications within PubMed. You need to figure out from the already available literature on CLL, on this disease, what is the percentage rate that is favorable or not favorable for a specific patient. I'll let you look into this, and if you have questions, we'll address it together. Then, for this specific sequence, the question is, where are they localized the mutations? And the answer is 0.9 of the table. Then we go to the second part. You'll work a bit on the junction, give the junction on amino acids. For the physicochemical properties of the different amino acids, I would like you that you know where to retrieve them, more or less. Please don't give me a report that will be like an encyclopedia with all the physicochemical properties. Then I ask you to tell me which are the 5 important amino acids. Here, I'll ask you to do a domain gap align, which is at the amino acid level. This we did not get to see it, I suppose. But it's straightforward. It was incorporated in the results of the quest. And then do an IMGT collier de Perle, the one that we saw. The domain gap align, which is equivalent to VQuest, with the difference that it's working with amino acids instead of nucleotides. It asks you to identify the alleles of the V and J gene that they are most closely related to your sequence. Give the percentage identity, have a look at the alignments, give the name and the position of the 5 conserved residues. IMGT collier de Perle. You'll give the description of the domains, which are the domains, the frameworks, and the CDRs. To identify the frameworks, the CDRs, which are the hypervariable regions, les ancres, what I told you, these are the squares, and the important residues that they are given in red. And at the end, there is a general question, like stepping a bit back. What is the purpose of these tools? What does IMGT VQuest do? What is IMGT domain gap align do? What does IMGT collier de Perle does? And a little conclusion on IMGT. So basically, I would like you to work on this homework. If you have any questions, we can either address it now or how do you want to work? Do you have any questions right now? Do you want to start working now and you'll be asking questions? Or I can let you work on it And then we are together again on Wednesday in the morning. We'll start with this one. We'll start with you showing the previous homework and this one. All together, we'll address on Wednesday these questions that you have done. And before we start on Wednesday, I would like to receive, should I receive it or should I not receive it? I would like to receive it, why not, your homework. To make sure that everyone will have done this homework. The one that you are supposed to have done today. If you have not done it, you have a last chance up until Wednesday to finish it. And you have the rest of today to work on your homework. And then we'll correct it on Wednesday. Is this okay?
Yes. So you mean that the next class we have to submit our homework like six exercise, right?
Yes, please.
Okay.
However, they are not 6. Don't tell me that I give you too much work to do and you start complaining. You were supposed to have done the first exercise today. I only want number 1 that you were supposed to have done today. And then 2 and 3, they are not exercises. There is 4 exercises that we went through together. There is 5 and 6. There are 3 more exercises that I give you now for Wednesday. Is this clear?
Yes, it's clear.
And I would like that you send it to me by 8 o'clock in the morning, French time, on Wednesday. And then we'll go through the different exercises and correct them together on Wednesday. Okay?
Okay.
Just can I ask what is
Of
the
course.
time in France now? The time, the time clock.
In France?
Yes. So I can calculate the deadline for us.
What time do we have a class on Wednesday? Just before you start your class? The idea is, before we start correcting your exercises, I would like to have received your exercises, your homework. So, we don't correct them and then all of you have given the correct answers. How will I know that you have done? I'm happy to have little mistakes done by you so I can see what was not clear and I can address it. If everything is correct, then I don't know what you know and what you missed. So just before we start the class, usually they start the class at 8:30. If you send it at 8:25, that's fine. Right now it's 10:15 more or less in France. In the morning.
Yes, it's okay.
Is this okay?
Afternoon for us.
Correct. So it's just before we start the class, the latest. If you have it earlier, send it to me. But please, don't start doing your exercises 5 minutes before you come to class. It will not work. You need more time to do your homework. Is this clear? Perfect.
Yes, it's clear.
In case you are completely lost somewhere and you don't know how to do your exercise, ask me a question. Do you have my email? You have my email? Ask me a question. Don't wait up until Wednesday to ask me the question saying, I did not understand this at all. Okay?
Yes.
Perfect. So I'll let you start your homework.
Okay.
Yes.
Thank you very much. And I'll see you on Wednesday.
Yes. See you on
See you on
Wednesday.
Wednesday.
Take care. Bye-bye.
Bye bye.